Okay, so I'm going to record this for our colleagues who aren't here today. Our first lecture here really is about pharmacology. We'll go through a lot about drugs and FDA, a lot of information in this lecture. Much of this comes out of chapter 51 of your text, which I urge you to go ahead and read. We'll talk about a lot of pharmacology first, which is the study of drugs, the science and the science of drugs action in biological systems. So let's define a drug. There are lots of definitions of drug. For the purposes of this class and for today, we're going to define it as a chemical compound that's used to prevent, diagnose, or treat a disease or an abnormal condition. There are lots of ways to define drugs. We talked about a different definition of drug in COM 103 a couple of mods ago. This is going to suffice for what we're working with now. The role of the MA in pharmacology is to understand a few things. Medication errors can cause injury or even death. Many medication errors occur because of simple errors, simple mistakes. The answers can be, the errors can arise from prescribing the wrong drug. There are many drugs that have similar names. So prescribing the wrong drug, prescribing the wrong amount, prescribing the wrong route of administration. You know, there are many routes of administration. Most of the drugs that you're most familiar with, we swallow. We call that oral administration. Other drugs can be ingested, or injected rather, which is what we'll learn about here. Other drugs can go on the topical part of the skin and be absorbed into the body that way. So there are routes of administration. Fortunately, most of these get caught, these errors get caught before the patient is exposed to something that is potentially dangerous to them. And they get caught because we have mechanisms for checking at every step along the way. We'll learn about those as well. The other thing is that you need to be able to answer basic questions about the drugs and to educate the patient. Now, this is different from counseling the patient. Counseling the patient is really the doctor or the PA or the NP's job, okay? But if the patient says to you, well, how do I take this drug? Or what's this drug for? Because a lot of times they won't ask the doctor that question. Drives you nuts. But a lot of times they won't. So we need to be able to answer some basic questions. We need to be able to translate prescriptions. We did that in COM 103, so there'll be a little bit of review of that. You saw how bad some of those prescriptions can be. It's really kind of interesting. The physician practice that we use, all of their prescriptions are generated by the computer. So the doctor will type in what they want and hand you a written or a printed copy, much more readable. Things are improving quite a bit. But we also need to follow our legal requirements. Legal requirements including there's my buddy again, scam likely. And I apologize for not turning the phone off. I'm expecting a phone call about my father sometime this morning. We need to be able to keep accurate records. The legal requirements are described in the state under the Department of Regulatory Activities, known as DORA, and describe the scope of practice that an MA is allowed to engage in. And so you're required to know that as well. We need to be, when we are interviewing our patients, we are confirming what medications they are taking. And that includes everything. It includes all over-the-counter meds, supplements, naturopathic or homeopathic medications they may be taking. All of these things can interact with drugs in sometimes expected, sometimes unexpected ways. Those interactions can be very dangerous for the patient. So we need to know what we're taking, all of these things. We need to know if the patient is using alcohol or recreational drugs. 90% of the time or more that you ask that question, the answer is going to be no. A goodly proportion of the time, that answer will not be truthful. So the best thing we can do is do a little probing. My favorite question for alcohol use is, how many beers or drinks do you have in a day? Okay. That requires, and most people are, if they drink, will give you an answer. That answer will probably be minimized, but it will tell you that they use alcohol. Recreational drugs are a little harder. In this state now, we can say, do you use marijuana? Because it's legal, right? And a lot of times people will just admit that they do. Other drugs, it's harder to get them to do that. But we do our best with that. And we assist in patient education about drugs. And part of that is just essentially it's how you take the drug. As I say, we don't counsel the patient about what to expect. That should be the doctor's job. And you should make sure that that's taken care of. We also administer drugs. Again, we look at the state regulations and our scope of practice. Here in Colorado, the MA can give injections. The MA, some IVs, not in general. So bear that in mind. When an IV is to be administered, usually it's going to be a nurse that's associated with a practice that you're in. But the scope of practice allows that. It also allows you to draw blood. It allows you to do finger stick tests, like testing for glucose or testing for clotting factors, what we call the INR, the International Normalized Ratio, also known as ProTime, prothrombin time, for clotting function. We do that with patients who are taking anticoagulants like Coumadin. To understand the basic principles of pharmacology, you need to understand the basics of these things. And of course, translating prescriptions, checking and calculating doses. Doses vary. Many drugs are given based on the weight of the patient, based on the age of the patient. So we need to know how to just make sure that the proper dose is being given. Even oral drugs, we want to be sure that if the dose is supposed to be 1 capsule, that's all we're giving to the patient. And that inadvertently we haven't loaded 2 in. Or if we're giving multiple meds, we want to be sure that the proper number is in each dosing container. So we want to be able to do that as well. We need to be able to answer these basic patient questions. When should I take it? Should I take it with meals? Should I take it, how soon before I go to bed should I take a drug that's going to help me sleep? Well, you don't want to take it 2 hours before going to bed because you're driving home, right? So you want to be very careful about that. And of course, we're going to be emphasizing adhering to legal requirements, and that is dependent on your jurisdiction. Here in Colorado, our scope of practice might be different than if you say move to Kansas. You need to know what the scope of practice rules are where you are. Your MA certification, when you get it, is a national certification, so that you can transfer that and use that if you decide you're going to go live in. I've had a number of students, I don't know why, but decide they're going to go live in Texas. Well, they have a national certification that allows them to transfer their skills into that environment, but they still need to understand the scope of practice rules. Now, where do drugs come from? Well, plants. We've used plants. If we go back into history, every group of humans had a person. We sometimes call them shaman. We sometimes call them the medicine person, medicine man, medicine woman. These are people whose job it was to treat injuries and disease. And plants and minerals are some of the oldest that we have. We'll get to the animal part in a minute, but we use these drugs. Digitalis, I mentioned earlier, Digitalis purpura, the source of our digoxin. Morphine comes from the poppy, the opium poppy, and among other things, Cenapod derivatives are used for constipation. If we want to dilate eyes, we use a derivative of deadly nightshade, belladonna. We have lots and lots. In fact, for many years, there's some stories about actually Charles Darwin's grandfather, Erasmus, was a physician. And he was one of the leading physicians of his day. And he was founder of what evolved into what is now the Royal Society in England. He used to go through the countryside and visit villages and find the local medicine woman, usually a medicine woman, in the village, and ask what she used for treating disease. And so if she was willing, she would tell him. He would go collect a sample. He always traveled with a great big case full of little drawers to collect samples, and he'd collect the samples, and he'd go back to London, and he'd try them out in his practice. If they worked, he'd start using them. If they didn't, he'd throw them away. So that was how we found drugs. It was trial and error. And how did we find it? How do we know what plants to try? Well, we often watched what animals did when they were ill. We would look at the drugs, at the sources of animals that we're using, or where animals would lick, where they would go and eat things when they were obviously ill. And then it was a case of trying. Sometimes the results were less than what we wanted because, of course, animals and humans are different. But that's one of the ways we did this. But this was all what we called empirical medicine, meaning it was experimental. And so doctors would come back, they would let their colleagues know when they found something successful, and it would go from there. Animals are also were a big source. Chinese medicine uses various animal parts in a form of medicine that's based on similarities to treat human condition. But more recently, we've used a lot of sources. The original sources of insulin were beef and pork pancreas, insulin being produced in the islet cells on the pancreas. we were isolating that. That started in the early part of the 20th century in the treatment of insulin-dependent diabetes. Thyroid disease was treated by isolating thyroxine from pig and sheep thyroid, other animal thyroid as well. Insulin can be, by the way, isolated from a number of different animal sources because it's what we call a highly conserved protein, meaning that many species have very similar forms of insulin and can be transferred. Minerals, we've used iron for treating iron deficiency anemias. Iodine we used as an antiseptic, and zinc compounds, zinc oxide you're familiar with, can be used as a sunscreen. Iron and iodine are both very toxic, even though we require them. The amounts that we have, these are trace elements. Remember, in COM 103, we talked about total parenteral nutrition, and I talked about trace elements. Iron is one of those. Even though we think we have a lot of it, because of course we have 5 pints of blood, we don't. We don't have very much at all. Iodine is used also as critical for thyroid function. And too much iodine can actually damage the thyroid. So these are small amounts, but we have used them for years. Other sources of drugs, many of our antibiotics come from microorganisms, usually molds and fungi. The penicillin is not chemically synthesized. It's too complex and too delicate a molecule. So we isolate that from some specific strains of mold. Penicillium chrysogenicum produces our oral form of penicillin, potassium penicillin G. We get a lot of things from microorganisms that are found in seawater. So we find these things that produce these very complex molecules. Synthetic or semi-synthetic. We use the term semi-synthetic because what we do is we isolate some natural products and chemically modify them. For instance, the original penicillin form could only be injected. It could not be taken orally. So we have done some chemical modifications on it to produce things like amoxicillin, which can be taken orally. And we stabilize those by using some chemical processing. That's what we mean by semi-synthetic. Most of our drugs are synthesized these days from basic components. They're completely synthetic. In origin, we make them we do not involve isolating them from a natural source. The morphine derivatives, oxycodone, and hydrocodone, and those are semi-synthetic forms of morphine and codeine. So, and more recently, we've used advanced biochemical manufacturing. We call it recombinant DNA techniques. We use cellular sources. Originally, we used bacteria. Now, many of our drugs come out of human cells, such as insulin, which is now the drug Humulin, a brand name for a short-acting insulin form, is produced in human derived cells. So These are viewed as safer. In many cases, they appear to have basically a different safety profile. You can get too much of a good thing. This is a picture of the flower of the foxglove plant, it's a beautiful flower, isn't it? Which we use as our source of digoxin. This is a picture of penicillin mold growing, on a plate. Penicillin was actually discovered by accident. A microbiology lab in London, run by a Scottish microbiologist by the name of Alexander Fleming, was experimenting with studying the growth of bacteria. And in England, they don't have screens on windows. They don't have that many insects. That's a blessing and not so much of a blessing sometimes. But they don't have screens. And so it was a hot summer day. The laboratory windows were open. The laboratory next door was looking at the growth of a different microorganism type, molds. And so what happened is some spores of bread mold blew out of the laboratory next door into Fleming's lab and settled on their petri dishes. They didn't cover them. We do now. They didn't cover them to keep them from being contaminated. And so Fleming was gone for a few days. Graduate students came in and found their petri dishes contaminated and tossed them away. Fleming came back, said, what happened to them? Oh, they got contaminated, and we threw them away. Well, Fleming was a good scientist. He said, I want to take a look at them. And what he noticed was that where this mold was growing, there was an area of the petri dish that the bacteria wouldn't grow in. And he immediately understood that the mold was secreting something that prevented bacterial growth. And that was his realization was that it might be able to be used to treat bacterial infections, which were a plague. We had no real drugs for bacterial infections at that time. So that's how that happened. So we won't be doing this. This first question here is, medical assistant needs to be sure that the physician is aware of all medications that the patient is taking, and needs to be able to ask patients about alcohol and drug use, and educate the patient. Okay, and they also may be responsible under Practice Act, and it is here in Colorado, that they can administer some medications. Now, pharmacology is divided into a number of different subdisciplines. Pharmacodynamics, we'll get to each of those. Pharmacodynamics, pharmacokinetics, pharmacognosy, toxicology, and pharmacotherapeutics. Of those, pharmacotherapeutics is probably the most obvious definition, using drugs to treat disease, right? So let's go and look at what some of them are. So pharmacology, pharmacognosy is natural products, study of plant and animal and mineral sources. So the characteristics of those drugs. They can be complicated. Digoxin regulates the heart. In higher doses, digoxin actually will stop the heart. So we need to be able to understand that natural drugs are very real. They're very potent. And they are also very dangerous in many cases. So we need to understand them. You'll find that with most natural products, the doses are very small. We talked about doses in COM 103 in milligrams. For many of our natural products, we're talking about microgram amounts, 1,000-fold smaller. Pharmacodynamics is the study of what drugs do to the body. In other words, how does the drug affect its therapeutic action. Okay, what the drugs do to the body. Pharmacokinetics is the study of the reverse, what the body does to the drugs. And we'll get to what that means in a bit, but the body doesn't like foreign substances and does its best to get rid of them. And how it does that is the study of pharmacokinetics. How and how fast. Pharmacotherapeutics is obviously how drugs are used to treat disease, both singularly and in multiple settings. Toxicology is the study of how drugs negatively affect, how they is looking at poisons and the poisonous effects of drugs. And those poisons can be natural products coming out of pharmacognosy. Famous story of Socrates being told to commit suicide by drinking hemlock. Hemlock has a very toxic chemical in it that will kill you. And there are many examples of those. So it also looks at the interactions of drugs. Drugs interact with each other. They interact with food in different ways. And so toxicology and pharmacotherapeutics together study those interactions and the effects they have on the body. Those interactions can be multitude. We'll get to some of those later. And the adverse effects, the actual, what is it that we see? Does the drug affect the kidney? Does it affect the functioning of the heart? Where do they have their effect? So if we look at the difference between pharmacokinetics, PK, and pharmacodynamics, PD, we have a relationship. Pharmacodynamics, we saw, was what the drug does to the body. Pharmacokinetics is what the body does to the drug. And there are a number of different steps in what the body does to any drug. First off, it has to be absorbed. If we are giving a drug by a route, it takes some amount of time to absorb into the body. The only time that time is literally within 1 to 2 minutes is if we give the drug IV. Then it's absorbed into the bloodstream. It's injected there. It still has to then, after it's absorbed into the bloodstream, it has to go somewhere. And that is the process of distribution. Where in the body does it go? Depending on the route of administration, it will go to different places. If we swallow a drug, it has to be absorbed from our stomach. The first place it goes is to the liver when it's absorbed from the stomach. The liver is the main metabolic organism. So this is metabolism. Metabolism is changing the drug so that it's easier for the body to now eliminate it. Okay? When the drug is taken orally, the first distribution is to the liver, where some of that metabolism occurs, but not all. The entire drug amount is not going to be metabolized immediately. So anywhere from 20 to 80% of that drug will be metabolized the first time it goes through the liver. We call that a first pass effect, going through the liver after oral absorption. Then what's left is back into the bloodstream to distribute to other tissues in the body. What other tissues? Partially that depends on the drug. We have mechanisms of directing drugs to certain kinds of tissues. We have tissues that will preferentially absorb certain kinds of drugs, but often they're just broadly distributed. And finally, and then it goes back into the liver again, further metabolized, and then it's eliminated. It can be eliminated through several mechanisms. It can be eliminated through solid waste elimination, or through the kidney, and then liquid waste elimination. Rarely it can be exhaled. We have certain drugs that that I've run into that people take them and they smell like they have just eaten about 17 pounds of garlic, because there's just this really garlic-like odor that comes out not only from their breath as a gas, but it actually will excrete through the skin. Rare. The vast majority of drugs go out through the kidney, or they're excreted into the colon and then passed in fecal matter. Most of them go through the kidney. That's why when you're asked to do a drug test, they ask for a urine sample. They don't ask for a fecal sample. They look for a urine sample. So the drugs, we look at what the drug does to the body. We have a number of different classes of drugs that act on different parts of the body. Drugs interfere or interact with cellular processes and biochemical processes. And various drugs will interact with various different ones. And we characterize that by looking at exactly where in the body, what body biochemical systems are being dealt with by this drug. And we use that as a way to characterize and classify drugs. For instance, aspirin and ibuprofen function by interfering with a pathway, in a cellular pathway in inflammation that involves an enzyme called cyclooxygenase. They're cyclooxygenase inhibitors. We call them non-steroidal anti-inflammatory drugs. And that's an old term because the mechanism of action of aspirin was not elucidated until a century after it was discovered and marketed. It was the 1980s. It was developed in the late 1970s in Germany, and it was the middle 1980s before a couple of pharmacologists figured out, because we now had the tools and the ability to do these studies, figured out how aspirin works. It got them both the Nobel Prize. How the pharmacodynamics looks at how the drug works to produce its effect, and it looks at the interactions between the drugs and the tissues and cells that the body has, where it is, and then the response that happens. To go back to aspirin, when we inhibit cyclooxygenase, we inhibit a pathway that leads to the generation of what we call inflammatory mediators, other compounds that raise tissue and general body inflammation. And so that's the body's response, is to not generate those mediators. So that's what that is related to. Another one is looking at drugs used to treat high blood pressure, known as hypertension. And so what we do is we study how effective antihypertensive drugs, by one of 5 different mechanisms, can be in reducing blood pressure. Okay. Obviously, another example is decrease in blood glucose levels on dosing with insulin. So we study, we look at that. That is the pharmacodynamics, the body's response to the interaction of insulin on the glucose receptors. Okay? And then I mentioned international normalized ratio, ProTime, during Coumadin. Coumadin is the brand name for warfarin. Coumadin therapy. Okay. Pharmacokinetics on the other hand, is the other way around. What does the body do to the drug? Not the response of the body to the drug, but what the body does to change the drug, to make use of it. And so we look at absorption, we talked about that, absorption, distribution, metabolism, and excretion. We call this ADME or ADME because we're lazy. ADME's 2 syllables. So we call it that. We also look at this in a lot of different ways, but we have to study it. Absorption depends on the route. There are slower methods. The slowest method of absorption is oral, swallowing a drug. That is the slowest mechanism of absorption. The fastest mechanism of absorption is to give an IV administration directly into the bloodstream. Okay. Then we have intermediate routes, intramuscular and subcutaneous routes of administration. Generally, we'll see absorption in about 5 to 10 minutes out of these tissues, as opposed to generally 30-ish from oral. There are also what appear to be oral mechanisms of dosing. Sublingual, which means putting something under the tongue and just holding it there and letting it dissolve. And buccal, which is between cheek and gum and holding it there, letting it dissolve. What happens there is, and suppositories, rectal specifically, or even vaginal suppositories, go into this tissue. We let the drug absorb through those tissues. They go straight into the bloodstream. The time for action is generally just a couple of minutes from those. We should start seeing the effect. One of the most common drugs given sublingually, for instance, is nitroglycerin, which treats the very painful condition known as angina, which is often a precursor to a heart attack. But angina is a blockage of arterial blood flow in the heart, which then results in pain. We put a nitroglycerin tablet under the tongue. It's absorbed directly into the oral mucosa, goes directly into circulation, and relieves the pain within a couple of minutes. If we had swallowed that, then even if the stomach hadn't destroyed the nitroglycerin, it would take half an hour. Nobody wants to be in that kind of pain for half an hour. So we have that route. So time to absorption. Distribution, basically it goes, as I said, true oral, swallowing a drug, the distribution goes through the liver first, and we have that first pass metabolism, which is part of the reason that it takes a half an hour to see the effects of an oral drug, and then excretion. Why is it important to know? Well, sometimes we need to tell the patients they have to take the drug on an empty stomach, or with water, or after eating. For instance, the antibiotic tetracycline needs to be taken on a stomach with food, because it needs acid to be absorbed. If you take it on a fasting stomach, it won't absorb. Okay. So it's now time for our second break. So I'm going to pause this recording, come back to it later. That's right. So we're back to the lecture. We've just been in the lab learning our first injection type, which was intradermal. We now have 2 experts in the class who can teach everybody else how to do this. So back to pharmacology, we have, again, a little bit more detail about the ADME process. Remember, absorption, distribution, metabolism, and excretion. So absorption is the process of getting the drug into the body and can help make it possible for the body to use it. The rate, as we said, depends on the route of administration. Oral meds have to be take about 30 minutes. That's why you take an ibuprofen. It takes about 30 minutes to get relief from your headache. IV is the fastest, goes straight into the bloodstream. All other routes are an intermediate amount of time. It also depends on the chemical characteristics of the drug. Some things need a little aid. Distribution is that transportation to the site of administration or the site of action. Metabolism is how we modify the drug to make it easy to get rid of. Remember I said the body hates foreign substances, wants to get rid of them. And drugs are foreign substances. So we usually want to find a way to get rid of them. Metabolism usually is a liver issue, but it can be in the kidneys. This is one of the major differences with the age of our patients and genetics of our patients, which is that older and very younger patients metabolize drugs differently than patients in that sort of mid-range 15 to 55 age group. Genetics, different genetics can affect the way drugs are metabolized and treated. And then actually the chemical characteristics of the drug itself. Most of the time, these things are modified in the liver and then excreted. They either can be put back into circulation for excretion via the kidney for the soluble, water-soluble metabolites, or if they're more fat-soluble, they'll be put out into the intestinal tract via bile. Elimination, most of them are eliminated by the kidneys. Some is the next most common is through feces, and then everything else, sweat, saliva, going out as bile itself, or exhaled air, or through breast milk in lactating mothers. That's why you will often see a warning about a drug not to take it if you are lactating, because then you're dosing your baby. Don't want to do that. So that's that process. So there's all sorts of things. Absorption is through the stomach, can be through the mouth, can be through the small bowel. We can have it rectally absorbed through suppositories or vaginally through suppositories, and directly across the skin in transdermal form. Distribution is through the entire circulatory system, including capillaries, and via the capillaries, it crosses then, where appropriate, it will cross the cell membranes. Metabolism, mainly the liver, and excretion, mainly the kidneys. So here's just a graphic that looks at what happens when we administer a dose. We get plasma concentration and then pharmacokinetic activities here can lead to elimination. Pharmacokinetics also through that metabolism often helps get it to the site of action. And then the pharmacodynamic effects, in other words, what the drug is doing to the body, can occur at that point. Okay. There's a couple of questions. Caring for a patient who has diabetes complicated by kidney disease. You need to make a detailed assessment when administering medications because the client may experience problems with what if he has kidney disease? Pardon? What's the main function of the kidney as far as pharmacodynamics is concerned? Well, it eliminates, right? Okay, so if you have kidney problems, you may have trouble with excretion. Okay, so that is something that has to be taken into account. That's why some drugs we have warnings about um, patients with kidney disease. Okay, so that's one. Okay, all right. All right, so let's take a break here. This will be a good place to pick up on Wednesday. So I'm going to escape out of this, and then we will. I want to talk about the research project and get you guys thinking about that right away. And before you leave, I have this contract for you to sign. So the research project isn't going to be writing a paper. It will be doing a PowerPoint presentation and that will include 10 slides, a reference page for a total of 11 slides. Needs to, when I say correlate with what we're studying in this class, topics can range all over the map. I've had people do a study of the development, the history of syringe type drug administration. It really goes back almost to ancient times. We've done things like that. So we've talked about that. Maybe the development of the modern syringe, maybe looking at the issues related to why you would choose a certain route of administration for some drugs and why some drugs have to be administered by injection and others can be administered by other routes of administration. So something related. You might actually do a little more work and understand some of the details of drug metabolism. You know, we're not. So it's going to be something you'll present it in class the last week of class. And again, remember you're dealing with friends, so nobody's going to bite. And these presentations are always pretty benign. But pick something that's of interest to you. And one thing you may do is just take a quick look at, we're reading chapters 51, 52, and 53. And in the last week of class, we go back and look at chapter 43, which is related to ophthalmology and working in an ophthalmologist's office. Take a quick look at those chapters, pick a topic. And then I'd like you to just let me know what you want to do. Send me an email, tell me in class on Wednesday, whatever. Tell me what you'd like to do as a research topic. And then we'll have time in class for you to do that. So it'll be due on the last week of class, which is, it'll be due on Monday, the 29th of July, before class starts. So by the time class is done, you might want to aim for getting it done over that previous weekend. And then in week 4, you'll find a place in the week 4 module to submit the presentation. Okay, and then we can run it probably just from my computer. Since I'll have a copy of it. So you'll make a presentation to the class. Presentation of a 10-slide PowerPoint probably shouldn't take certainly any more than 10 minutes. So it's not we're not expecting a long lecture here. So that's what I want you to be working on in this class. Let's see, let's back up and take a quick look at this week's assignment. Now, Wednesday, I want to spend some time looking at the vaccine war video. But in the meantime, take a look at these two videos here. They're not terribly long. I think one's about 15 minutes and the other's about 3 or 4. So, and you're going to write a short discussion of what you learned on that and include what you've learned watching the vaccine war video. Okay, so we'll watch that in class on Wednesday. That'll be about an hour long. So we'll have that activity on Wednesday as well as finish off this lecture. So that's the assignment for this week. Again, the discussion, how do you feel about needles, okay? Discuss how you feel about giving injections. Does the idea of giving injections bother you? And how might your feelings influence patients who are receiving those injections? Okay. Talk about that. The usual 150 plus words by Wednesday midnight, and then 2 responses by the weekend, by the end of the weekend. So I've started off, excuse me, I've started off the discussion, so it should be okay. All right. So, questions? We'll back up here to what else? I will go ahead and post the Zoom session as well if you want to look at that and have a review of what we talked about here. The assessment, I suggest you read chapter 51 and do the assessment and be prepared to, to get that done. These assessments can be done outside of class, except for the week 4 assessment. The last one, we'll do that in class, okay? But we'll do that. If you have questions, email me. I check email regularly. I'm here usually in the afternoons on Monday and Wednesday. So if you want to stay and talk about something, let me know. I'll be happy to do that. Okay. So other than that, I think we're done for the day.